We're now ready to begin a new unit, although it is in effect just the second part of what we have been doing. We've been dealing with heredity, and so we've done heredity 1, and now we are ready to begin heredity 2. Before we really get into anything specifically, let's just get a general sense of where we've been and where we're headed. We started off the year learning some basic biochemistry and some chemicals. Those chemicals were important because they composed cells, which you see pictured here. When we learned about cells, we learned about all the organelles and the specific functions within, and one of the parts that we learned within the cell was the nucleus, pictured here. The reason the nucleus is so important is because it houses our chromosomes, pictured here. So we're getting smaller and smaller and smaller as we go through these pictures. We've just spent the last few weeks dealing with different traits and how they're passed down from one generation to the next, and those traits are housed within these chromosomes. What we're now headed to, and what the main focus of this new unit is, would be the makeup of these chromosomes. In other words, we need to talk about DNA and specifically how it functions and relates to the heredity and those traits that we've been passing down. In terms of DNA, there are several big names from a historical standpoint. Probably the two most notable would be here on the left, James Watson and Francis Crick, in that they are given a large portion of the credit for discovering the basic structure of DNA. The reason that that's so important is because you really can't understand how something works until you understand what it's made of. And based on what I just showed you in the previous picture, we're talking about something that is tiny. The biggest thing I showed you in the previous slide was a cell, and that is microscopic. So if you go down several levels from there, you can imagine how small DNA must be. And so to understand its structure had to precede understanding what it does, understanding its function. And Watson and Crick, as I said, are given the lion's share of the credit for discovering the structure of DNA. Maurice Wilkins and Rosalind Franklin also played a large role in that, and we will talk a little bit about that at a later date. Watson and Crick discovered this structure, or figured out this structure, back in 1953, and going into it, they had some basic background information that others had figured out before them. They knew that DNA was a long, thin molecule. They knew that it had 4 nitrogen-carrying bases, or sometimes just called bases or nitrogenous bases. But absolutely positively, these 4 names right here of nitrogen-carrying bases are 4 things that you have absolutely got to know: adenine, guanine, thymine, and cytosine. In addition to those 4 parts, they also knew that DNA contained what's called a phosphate group, also a 5-carbon sugar called deoxyribose, which is in part how DNA gets its name. DNA stands for deoxyribonucleic acid, and so that D of DNA comes from the sugar that composes it. 2 other interesting things that Watson and Crick were made aware of was that the amount of guanine in any one DNA molecule was always, for some reason, equal to the amount of cytosine. And the exact same thing could be said between the relationship of adenine and thymine. So basically, Watson and Crick and Franklin and Wilkins knew these 6 things about DNA, and, through the help of Rosalind Franklin, had this picture that you see over to the right. Now you think, okay, well, there's a picture. How hard could it be to figure out the structure? Well, you can imagine that looking at this particular picture, taken through a process called X-ray crystallography, not all that helpful. So Watson and Crick were off to figure out, in 1953, the basic structure of DNA. A little bit more about those all-important nitrogen-carrying bases that I mentioned. Here you see pictured 2 of them, adenine and guanine. The reason that they're pictured together is because they have very similar structures to each other. And because they do, they are lumped together in a category of nitrogen-carrying bases called purines. you can see that the commonality that they have is something referred to as a double ring structure. Here's one ring, here's another, and you can see the same basic structure with guanine. The other two nitrogen-carrying bases, cytosine and thymine, are similar enough that they fit into the same category of being called pyrimidines. And the similarity between them is that they lack a double ring structure, but instead both have a single ring structure. What Watson and Crick figured out is that the reason thymine and adenine were always in the same quantities is because they were always attached to each other. And you can see that pictured here. The amount of thymine and the amount of adenine in any one person's DNA was equal, because wherever there was one thymine, there was always one adenine attached to it. You can see the dotted lines here and here. Those represent what are called hydrogen bonds, and we learned about them back when we talked about water. And so the amounts of those two nitrogen-carrying bases are always going to be equal, regardless of who we're talking about. The same could also be said of cytosine and guanine. Their quantities were always equal, because wherever there was a cytosine, there was a guanine attached to it. You can see all the dotted lines. There happen to be 3 hydrogen bonds connecting every cytosine and every guanine. In addition, the other two parts that we talked about, deoxyribose, pictured here on the left, a 5-carbon sugar, and then there is also phosphate. And if you look here at the diagram to the right, the connection Watson and Crick figured out there is that there are alternating quantities of them. In other words, here is deoxyribose, here is phosphate, here is deoxyribose, here is phosphate. They form an alternating pattern of sugar, phosphate, sugar, phosphate, sugar, phosphate. And eventually, what Watson and Crick put together is this basic structure that you see here. Here is a phosphate, sugar, phosphate, sugar, alternating back and forth the way I just explained. And there are 2 sets of those. So one here on the left, one here on the right. And each one of the sugars is then connected to one of those nitrogen-carrying bases. So in this case, thymine, which is always connected to an adenine. In this case, cytosine, which is always connected to a guanine. Notice that it looks very much like a ladder. We have the sugars and phosphates on the right and the left, making up the sides to the ladder. And then we have the rungs to the ladder always being 2 nitrogen-carrying bases, always having an A with a T or a C with a G. These, the nitrogen-carrying bases, are referred to as base pairs. So the rungs of the ladder are always made up of base pairs. The other thing that's a little bit unique about the structure here that needs to be pointed out is getting back to the sugars and the phosphates on the right and the left. Notice that they are running in opposite directions. This particular sugar up here is numbered. Carbon number 1, carbon number 2, number 3, number 4, number 5. Notice that it's the 5th carbon that's sticking up down there, whereas it is the opposite here. We've got 1, 2, and it's the 3rd carbon that is sticking up on this side. And so these two run in opposite directions. The terminology that we would use for that is anti-parallel. This one runs in a 5' to 3' direction, and this one is running from a 3' to a 5' direction. It would be as if you laid 2 people down end to end, or side by side, I guess I should say, but you faced them in the opposite directions. They're lying parallel to each other, but they're lying parallel in the opposite direction. In other words, they are anti-parallel. And that's how a DNA molecule is put together. And if you take that ladder structure and you twist it, you get what DNA really looks like. The phrase that Watson and Crick coined to describe the overall structure of DNA is that of a double helix. A helix is a twisting structure, but because this helix has two sides, they refer to it as a double helix. So the sugars and phosphates on each side are then connected by the nitrogen-carrying bases in between, the rungs to the ladder, and you can see everywhere there is a C, there is a G joined to it. Everywhere there is an A, there is a T joined to it. Notice that those rungs only extend from the sugars. In this particular picture, the blue circles, the blue spheres, they are the phosphate. There's nothing coming off the phosphate. See it here, see it here. The rungs to the ladder extend only from the sugars, and that's what's represented by the gray structures here and here. And that gives you the basic overall structure of DNA. 6 parts. 4 of them are the nitrogen-carrying bases of cytosine, guanine, adenine, and thymine. And then the other 2 making up the sides of the ladder, of the twisted ladder, would be the deoxyribose, which is a sugar, and phosphate.